119 million metric tons. That's how much nitrogen fertilizer is projected to be used just this year. And the energy used to make that fertilizer is only slightly less than what we'll use to light all of the world's homes. Unfortunately, as much as 50% of the nitrogen applied to a field will wash away rather than being absorbed by crops. This extra nitrogen pollutes groundwater and sets off harmful algal blooms. So why do we use so much? Crops often need more nitrogen than is available in soils, and the 78% of our atmosphere that is nitrogen is in a form most life can't use. So humans use an industrial process to fix nitrogen, that is, to take it from the atmosphere and turn it into a useful form. But there are also bacteria that can naturally do this, and certain plants recruit such bacteria to live inside them, thereby getting direct access to just the right amount of that useful nitrogen. Farmers have long been taking advantage of this by rotating crops with legumes like clover, so the bacteria in their roots, called rhizobia, can refresh soil nitrogen. To figure out if we might be able to get other crops to recruit their own fertilizing bacteria, we first need to understand how plants naturally form these partnerships. So far, attempts to engineer the legume-rhizobium relationship into other plants have been unsuccessful. But some plants form an apparently simpler association with other nitrogen-fixing bacteria called cyanobacteria. And we think that by studying these cyanobacterial interactions, we may find easier ways to engineer self-fertilizing crops. One of the types of plants that forms these associations is hornworts, which was supposed to be behind me, but they are tiny plants smaller than your fingernail that have no flowers or seeds or plumbing to move water and nutrients around, but they do make good plant models in the lab. And in my current work, I'm surveying the cyanobacteria that live in hornworts around the world, and I've been excited to find that there's a broad diversity, including some species that might be new ones. By comparing these cyanobacteria to others that don't form plant associations, I hope to figure out what makes a particular cyanobacterium a good plant partner. I'm also helping to sequence some genomes for these partners in order to find the genetic basis of the relationship. And to study the functioning of this interaction, I'm combining various hornworts and cyanobacteria, and I've already found that some cyanobacteria are better at colonizing hornworts than others. The next step then is to see if that translates into better nitrogen transfer. And I hope that this work will lead to future engineering of similar relationships in crops that will help reduce our need for synthetic nitrogen fertilizers. Thank you.